Okay, and thank you for your introduction. The topic is my presentation is identification and global characterization of eccDNA reveals hallmarks in IR nanoparticle-treated breast cancer cells. Actual chromosomal circular DNA is eccDNA for short. Is circular DNA that read in size from hundreds of bases to megabases and found in both normal and tumor cells. Recent studies have discovered their ability to regulate gene expression and immune response, such as they can enhance genes expression or produce microRNA to suppress gene expression, or they can transcribe it. On the other hand, the origin of eccDNA has also been described several models. For example, the eccDNA produced may be related to the DNA damage repair or dying cells or transcription. But none of these models have explained the difference, the changes in eccDNA in the same cell lines, but under the different conditions. So we want to investigate the eccDNA profile and explore the association between the transcription and the eccDNA. We induced IR metabolism using IR nanoparticles, and then under the same concentration of the IR nanoparticles, we induced the ferroptosis using the gene interference method to test the characterization of eccDNA in dying cells. Since the transcription make an important role in the origin of the eccDNA and functions, we think co-extraction method was required. I think if eccDNA and RNA isolated using the distinct protocols, we are mix the eccDNA data may not match the RNA data, so co-extraction method was used in our studies from the same cell lines. However, to my surprise, the high heterogeneity was observed in our studies. We found only a few eccDNA with the identical junction site. Each replicate. Also, the number of common eccDNA with 19% overlap of sequence is increased, but is very still little compared to the total number of identified eccDNA. So we repeat some. We found this phenomenon in biological or technical repeaters in other cells. So we think the high heterogeneity of eccDNA may be posed a challenge for developing a specific eccDNA as a hallmark, because a specific of eccDNA may be not often identified in its replicate. So we suggest that using a specific eccDNA as a hallmark should be performed with great caution. So searching for a stable feature of eccDNA will focus on our studies. Since the biogenesis of the eccDNA is associated with transcription activation, we infer that the majority of eccDNA noted genes could overlap. Indeed, we found more than half of the eccDNA noted genes are overlapped in replicated. So we focus on the eccDNA noted genes to analyze. We can see that 7,000 overlapped eccDNA in cells with or without IO nanoparticle treated, and about 2,000 eccDNA noted genes are present only in IO nanoparticle treated cells. So we focus on this part of genes to analyze, and we call this part of genes is eccDG, and then we analyze the common we analyze the amount of the common eccDNA in DEG and eccDEG, and found a small part of eccDNA noted genes overlapped. I think this part of genes maybe reflect a specific state of the cells in response to environmental changes. Also, we found another normal breast cells had a similar results. We can see that a large number of eccDNA cannot always come from the DEG regions, and a lot of eccDNA seems unrelated to transcription. So we need to eliminate the background eccDNA and combine the other data such as transcription data to analyze the eccDNA characteristics. But I found an interesting phenomenon. I observed a high correlation in the distribution of the normalized eccDNA count across the chromosome in replicates, and the genomic elements distribution of eccDNA also showed a high correlation in replicates. So I think the global distribution pattern of eccDNA have stability. So we analyzed the chromosome and genomic elements distribution of eccDNA and found the eccDNA had a significant change on chromosome 16, 17, 19, and 20 in ion nanoparticle treated cells, which infer that the generation of eccDNA should have some hotspot regions. This phenomenon was also observed in replicates in different cells. As expected, the genomic elements distribution of eccDNA from the ion nanoparticle treated cells showed increase in untranslated regions and exon or CPG island regions, but in intron and intergenic regions was similar. This phenomenon was also observed in replicates. It has been reported that apoptosis have indeed increased the number of eccDNA. So I want to know whether ferroptosis have a similar characterization of eccDNA. So we induced ferroptosis using the gene interfered method, we call it shift method, to which knocking down the expression of two IO metabolic genes and then induced the ferroptosis with the same IO nanoparticle concentration. And then we found an interesting phenomenon. The chromosome distribution pattern of eccDNA in treated cells was similar to non-treated breast cancer cells. And the genomic element distribution of eccDNA were also compared to those of untreated cells. So we then analyzed the amount of common the genes between DEG and eccDEG, and found more than 3,000 unique eccDNA annotated genes, but only 147 DEGs in GIFT-treated cells. In contrast to IO nanoparticle-treated cells, GIFT-treated cells had fewer DEGs, but the eccDEGs increased. However, IO nanoparticle-treated cells showed a remarkable variation in eccDNA diffusion and DEGs compared to its without treated counterpart. So we think if when cells are able to regulate many more genes to resist the ferroptosis, there were not a large number of eccDNA produced by a specific transcription. So maybe maybe showed a different diffusion pattern compared to the eccDNA produced by transcription activation. So in summary, the high heterogeneity of the eccDNA poses a challenge for developing a specific eccDNA as a biomarker. However, in our study, we found the genomic and element distribution of the overall population of eccDNA are highly reproducible in each replicate. So the global distribution pattern of eccDNA could potentially serve as a hallmark to reflect different cell states. Oh, that's all. Thank you for your attention.
Thank you so much for the very interesting talk. So I would like to ask if there are any questions from the floor.
Hi, great talk. Actually, I think I would like to ask a more general question. So you've seen a lot of patterns with eccDNA expression and like distribution along the genome. Do you think it is sort of an effect of just transcription, or do you think it also sort of benefits certain cells, certain populations? Because I see some interesting genes within the eccDNA circles. Do you think they are sort of like very important biologically in these cells, for example?
I think eccDNA may be short, so they cannot always have a gene on it. I know eccDNA may be referred to longer extra chromosomal circular DNA, so they can help the entitled genes. So I think they can transcribe it. But eccDNA, I think maybe if eccDNA is enough now, sorry, I think they can be transcribed. But I know some research have report eccDNA can produce microRNA and can suppress some expression and this.
Okay, thank you.
Hi. In one of the slides, you showed the normalized curve of eccDNA, but I wanted to ask, do you also try to do some spike-ins plasmid during the extraction to have a control for quantitations?
Oh, sorry, I have not sparked in some secure DNA on it. So I think we can we can't we can't see eccDNA copy number. So we only we only know how many eccDNA, unique eccDNA I identified in it. So we don't quantification yet.
Thank you. Can I ask one more question? So you were trying to find specific eccDNA for the condition. I'm not surprised that it's difficult to find because some people think that eccDNA, they are produced randomly. But in your another slide, you say you're trying to find whether they are produced by apoptosis. So that's the group of people think that eccDNA are produced randomly. So I think your direction seems a bit in contradictions here, because you are looking for a specific one, and then you say
yes,
from apoptosis, which one is specific, one is random.
Yeah, it's a hard process.